Hello, everybody. Thank you for joining us today for our webinar. And today's webinar is on our Aura platform on particle and aggregate quantification and identification and full USP testing with our new Aura platform. And just a couple housekeeping items before we get going. If you have questions during the presentation, you can post them in the questions chat box that you will see on the right-hand side of your screen, and we'll answer those questions at the end of the webinar. I'm Rick Gordon, and I'm pleased to announce our speaker today, Bernardo Cordovez. And Bernardo is Halo Labs founder and chief science officer. Bernardo leads our application and business development teams at Halo Labs. He received his PhD in mechanical engineering from Cornell University, where he also did his postdoctoral work. And he has a long history of developing a wide range of scientific instruments in areas such as particle detection, analysis, drug delivery devices, microfluidics, nanophotonics, and bioenergy production. And with that, I will hand it over to Bernardo, and he can start the webinar. Thank you, Bernardo.
Thank you very much, Rick. Good morning and good afternoon and good evening to everybody. Thank you so much for joining. I'm very excited about today's webinar, and so let's get to it. So our mission as a company is for you to know your particles, and what we mean by that is that we want you to understand all of the subvisible particles in your formulations, whatever they might be, understand every single particle and actually what they are, and to do that in a way that is validatable and that yields important results from the earliest point in the formulation development all the way down to lot release. And then today we're going to talk a little bit about some of the more upstream work that this tool can do, and focus the first half of the talk on the USP788 method too. We'll remind everybody how the backgrounded memory imaging workflow works, and into sizing, counting, and some of the new capabilities that the Aura instrument enables. And we'll follow with some conclusions and then your questions. So many of you here are probably very familiar with USP788. It establishes the limit for lot release for subvisible particles. USP focuses on particles greater than 10 microns and greater than 25 microns. That's very important to know, and many of you might be thinking about USP787, focusing on biologics, but actually 788 is the parent of 787. Now we have some of the images here that capture the old style membrane microscopy that is set forth as USP788 method 2. So 788 has two methods that are approved. One of them is light obscuration, the most popular one. And then the original one was membrane microscopy. And you can see some images there. There's an empty graticule, right? And it was essentially a long and arduous process, right, for calibrating the manual membrane systems using a color contrast membrane, as you see in that inset on the image in the middle, with the particles looking very bright. And the assembly process for doing the filtration and imaging is actually quite painful. So as you might imagine, and we're not really surprised, light obscuration completely took over membrane microscopy because it's inhumanely painful to run. And I don't think there's any other word that can describe it. It takes a long time to calibrate. It's very manual, very error prone. The inter-user errors were over the error, because everybody's individually quantifying, individual particles there. Essentially until we came around, and I'll get more into that later. But I think it's very important for us to focus on what are some of the very key merits of membrane microscopy. And if you read the 788 chapter, USP actually recommends membrane microscopy over light obscuration when you're working with high viscosity samples, because these flow cells essentially, when you have a high viscosity syrupy type substance, they tend to clog and cause all sorts of artifacts and all sorts of wall effects. They also recommend it for working with low volume samples, because all fluidic systems, whether they're light obscurators or fluid imagers, they unfortunately have a lot of dead volume working against them. In our case, we sample 100% of the sample, and that is not a marketing claim, but the fact that we image the entire membrane and 100% of the particles are in focus. It's fast because, again, you measure all of the particles at once. It's reliable because everything is in focus and you're measuring it with high refractive index. There's no loss, there's no washing of these samples. So in general, we can do measurements with and you can make measurements with the Aura from the earliest of stages to the latest of stages with just one tool, because you can start from 5 microliters to several milliliters of sample. And as we'll end up in the end, the fact that these samples are now in a membrane means that you can subject them to additional analysis and get more insights. And that's the second half of the presentation here. But most importantly, Aura now complies with USP788. So as of about February of this year, the informational chapter that essentially adds all of the meat of the validation and the research behind how to properly conduct 788. Chapter 788 itself is only like a page and a half, but actually most of the knowledge content is on the informational chapter. And it now supports automation and image analysis, just like the Aura does. So now you can use Aura for this work. And we're happy to send you some more information provided by the USP. What this really means is that you can use the Aura for this work. There's really good guidelines there for your own systems qualification, and we're happy to help you through that process. Some quick takeaways are that many membrane types are acceptable. The original old school membrane microscopy uses color contrast membranes, but essentially what they're saying is that as long as your membrane has a pore size below a micron, which is the case of our membranes, it works. You can validate it against manual membrane microscopy, and our system allows you to do that if you choose to do that. But I think like the overall USP message is that, I know that you guys are super busy. One of the reasons why light obscurators are popular is that it's sort of a green button solution that you click it and you get the results and it's fast, whatever they might mean, right? But for good validation, the USP's message is do good science, right? And you guys are great scientists, so for validating the system, you have to just make sure that it's counting correctly, that it's sizing right, that linearity follows, etc. Right? So a lot of it rests on you. These are just guidelines, but ultimately you're the ones who are going to say what is good enough and what is safe. So going into the BMI workflow, most of you are familiar with how it works, and it's pretty simple. So we have these membrane plates. We image the membrane plates to get the background, then we load the samples, and then we measure them. And it's pretty quick. It's about a minute per measurement, and just, maybe like 20 seconds of prep time. And the workflow is as follows, as you guys are familiar with. We take a picture of the membrane before particles have been filtered on them. Then we load and filter the samples through our proprietary vacuum manifold. You can see these different colored droplets here to show you how that works. Then we apply a suction through our filter, and the particles are filtered through the membrane. And then we reinsert the plate back on the Aura system, and what you see now is essentially an image of the particles essentially superimposed over the membrane, right? They're lying over the membrane, but the fact that we imaged the membrane in advance allows us to essentially register those images and do a pixel by pixel subtraction to distinguish the topography of the membrane with the true particulate material. And not only that, but because this is done in a dry environment, it enables high refractive index measurements that allow for reliable results. And this is incredibly important for proteins, right? Because the refractive index, the way they look in water, is that they're virtually invisible. And therefore, light obscurators and flow imagers will tend to undersize and undercount these. So going back into what kind of data you can get from the system, as with standard Subvisible analyzer, you're going to be able to get size, shape, and morphology, and all sorts of different size-based parameters, from equivalent circular diameter to ferrite diameters, etc. We'll produce image galleries, right? That is individual particle info that is very specific and detailed information. One level up is your sample information. It's an individual well insight, and that's where you get your particle size distributions. And that's typically where LO and flow imagers are focused on at high level insights, right? So you got lots of small particles, lots of big particles, but those low throughput technologies, that's typically where they, for lack of a better word, they capitulate. We also have different ways of analyzing the data and visualizing it with amazing data visualization techniques. One of the things that we like the most about our system is, given its low volume and high throughput nature, is that we can get quick high level insights from an entire experiment's perspective, right? You can see how many IgGs you have there, how many ETFE particles are there, show that your negative controls work. You get very high level insights very, very quickly of what does your actual experiment and complete particle distribution looks like, not just in a single well, but for a whole plate or more. Back to what we were discussing previously, USP788 does make recommendations as into how these samples need to be run. One of the things that they recommend is to pre-wash the membrane. When you're working with a color contrast membrane, there tends to be a lot of, for lack of a better word, dirt in these membranes. That's not really the case with our membranes, but our system is compatible with just a pre-wash. Then you follow the protocol as normal, where you would background the plate, you would load the sample, and after you process your sample, the USP also recommends a post-wash, essentially rinsing the filtered sample on the membrane. Several of our customers do this already. Typically, the washes range anywhere between 1 and 5 times, depending the nature of the sample. But this is something very compatible and very much in line with what USP recommends. So you don't necessarily need to do this, but to make it look as close to what USP788 recommends, a pre-wash and a post-wash are essentially recommended to be included in your protocol. Now let's look at this quantitatively about what this means. So for example, I think one of the newer items here might be doing a pre-wash. And in general, a pre-wash will reduce the total number of counts, right? But honestly, a very small amount. And you can see the histograms on the right for the greater than 2, greater than 10, and greater than 25 micron bins don't have a wash, while the ones, the 3 bars in the left include a pre-wash. And you can see that those counts pretty much for the 2 and the 10 bins are lower. So they dropped by, say, about 200 to 300, particles per mil. So if your data hinges, and 200 or 300 particles per mil is about 5 particles on a membrane at 50 microliters, right? So if your USP data hinges on a single particle, we absolutely recommend you doing the pre-wash. For most sample ranking in more complex samples, this is probably not needed, again, just because the difference is quite small. And in addition, one of the things that ourselves and our users have seen is that if you pipette a highly viscous sample on a membrane that is wet, it can help with filtration and make that process a little bit easier. So in general, sure, we recommend pre-washing if you're doing USP788. Particularly if you're subjecting your criteria to the large volume criteria, which tends to be a little bit more strict and every particle counts. With the low volume criteria, I think more particulate content is essentially allowed per mil, because ultimately what USP is trying to say is they're limiting the total amount of undesired particulate material that gets intaken by a patient. If your volume is low, then in some ways that is a little bit more relaxed. One other advantage that we do here is compared to manual membrane microscopy is there's actually quite a bit on the chapter on how does cleaning these membranes look like. But honestly, those chapters were written decades ago, before there were even CMOS cameras in the way that we have them now. You can literally do a well by well subtraction, so you don't really have to worry too much about what was there in advance. So you can reference your wells and subtract, for example, your background water counts, either by picking a reference column or subtracting it against itself. So essentially every well becomes, for lack of a better word, its own sensor, and you can background whatever very small irregularities may have been there to begin with. So that's a very powerful new feature of the Aura system. So we're talking about the USP, so let's start with some sizing. The way we calibrate the system's digital resolution is with an ASTM universal calibration slide. And these are how most microscopic systems are calibrated. This allows us to set our digital resolution, how many pixels are necessary to measure a given size. And then there's our own proprietary signal processing algorithms for accurate particle detection. We verify that, in fact, we're measuring correctly by using bead standards. Typically, we're within half a micron of a given bead, and here we show a 15 to 10 and a 5 micron polystyrene microsphere standard. You'd have measured within that range. So it does perform accurate sizing on beads. Let's get into counting with backgrounded membrane imaging. So a key advantage, as I mentioned before, of the Aura over the Horizon system is that now we sample 100% of the membrane. So while before we sampled about 63%, the new system samples 100% of the sample. And that's really important for validation because you want to measure everything and not have to use extrapolation factors. For example, in the case of flow imagers, as I'll show later on, you're only about 20% of the sample is truly in focus, right? So in our case, 100% of the sample, lies within 1 to 20 microns from the surface, right? So every single particle is in focus, right? And every single particle is measured because it's captured on this membrane. So we do not need to apply any corrections, and we measure the absolute particle counts. Again, with the Horizon, we can't do that. And again, the other systems, as I'll show you later on, flow imagers, for example, cannot do that. And the reason for that is that with flow imagers and even light obscurators, you have some sort of flow cell or capillary, and they tend to have the size range. And the way optics works is that the particles that are going to be in focus are those that are in the beam waist, wherever there's the most photons, wherever the particles are focused. And that's just a function of the objectives that you use. And the higher magnification that you use, the less particles are going to be in focus, or the smaller your depth of focus. So even if you use the smallest of objectives, only about 55 microns are going to be able to be in focus. And for a 300 micron channel, where you're just using pressure-driven flow, you're only going to have 10 to 20% of those particles truly in focus, right? And you can try to play tricks on this, but physics are physics, right? So you can't really get around this. So not only do we have everything in focus, but on top of that, understanding that these protein aggregates are low refractive index contrast, and we truly want to measure everything, we have this high refractive index measurement methodology, which inherently has much higher refractive index contrast, tens of points higher than you can with flow imagers or light obscurators. And that's really important, again, for proteins and biologics. So as a result of that, is that while even some of these tools can use higher magnification objectives, our images are much more deterministic, right? So the contrast that we see with our tools, you can see captured in the BMI, measure these tools with much higher fidelity and detection fidelity in general than any kind of dynamic flow imager in the market, as has been published by Coriolis last year. So also very important is that this tool actually enables high counts linearity. Oftentimes users say, hey, I'm concerned about the fact that particles may land close to each other, but the fact is that a 5 millimeter square membrane, there's a lot of real estate for more particles to lie, and then the fluid mechanics takes care of the fact that particles will not land close to each other, as evidenced by the really large counts linearity range that we show. So you can work with very complex, highly particulated samples. And most importantly, what we really enable is the ability to work with very low volumes and all the way up to large volume. So here we have a case in which we have aggregated human IgG. And you can see that there's an average count hovering around 60,000 particles per mil. And on the x-axis, you see cumulative volume, right? So we have IgG aggregates pipetted at 50 microliters per well over an entire plate. So 50 times 96, that's 4.8 mils. And we're taking a rolling average of this number. And what you can see is that even at the very low volume, so an n equals 3, not only can we get pretty good CVs, but it's very much representative of the results that you get for processing an entire plate, right? So you can trust the low volume measurements, but you can also run larger ones. And again, to run this entire plate, it took about a minute per sample, so about an hour and a half total, and you get really, really nice statistics. In this case, this image, we actually reference the plate against itself running a water plate. There were only a couple of counts there, so the curve essentially looks the same. And we've tested this very carefully at very low volumes as well, and we can show that the dilution linearity follows, again, for human IgG aggregates, whether you're using 5 microliters per well or 10 microliters per well. So these are low volume measurements that you can trust. One practical application of that, and then I'll fall back into USP talk, is many of you here might be working with AAVs. And here's an example of different images, membrane images, of what different serotypes look like under different conditions. The top 3 conditions are 3 different serotypes, just basically pipetted from the vials. And then the ones below are these AAVs after they have been thermally shocked at 73 Celsius for 2 hours, which happens to be the melting temperature for AAV5. And essentially what this captures is that these particles, they may come already heavily particulated even at the neat sample level, but they have much and highly varying aggregation characteristics. So going into AAV, we can see that the unstressed samples display lower counts, as showed in that image on the left, but when you thermally stress them, right, the counts shoot up. And we've also characterized, for example, in the case of AAV2, it's stressed under several conditions, and we can see that the control has the lowest counts, definitely a lot of aggregation during thermal stress, and then increasing stress as a result of rotation and freeze thaw as compared to a control. Now, we only did this with 10 microliters to get high-level insights to then replicates of 30 microliters a well. And this is very important because basically until now, due to the very low volume requirements of samples such as AAV, people have essentially relied to extrapolate results from tools like differential scanning fluorimetry to predict the stability of these samples, because measuring the particulate content has so far been impossible. And actually what we've seen is that if you compare our results with what's predicted in the literature, there's essentially almost no correlation between DSF and BMI. So what the melting temperature predicts versus what the actual particulate content is, it's inherently different, right? So DSF measures how likely proteins are to unfold, while we actually measure, we don't predict anything, we just tell you how many particles are there for a given condition. So you don't really have to predict what you can actually measure. And when it comes to particles, the BMI provides the solution. So I'm going to, now that we gave it this low volume example, let's talk about a little bit more about validation, right? And some of you may have encountered this. And when you're working with beads, right? Beads on membranes, they tend to aggregate. That is a feature of the fact that these particles will roll in a membrane, they'll find each other, and they'll stick to due to van der Waals forces. And while that's an undesired effect, at the end of the day, you do have the same amount of mass there. And think about it this way, what USP really cares about is the total mass, the total undesired subvisible, the amount of junk essentially that is being injected. You want to limit that, right? Particle distributions give you greater insights on that, but fundamentally, you just want to limit how much material gets in there. So what we can do is, because we have all of this information here, is, well, let's figure out how many counts we have by summing the total area of the detected material, and then divide by the area of a singlet, right? So and again, we do this for beads because beads tend to collapse on each other. And basically what we find is that we have a very smart expression engine software that allows us to run those calculations very, very quickly. And we can see that using these USP grade beads, we measure basically exactly what the bottle predicts, which is 3,000 particles per mil, with a plus minus 10% uncertainty ratio. So not only that, but you can see that you get really good data even at the low volumes. Typically, we run about 8 wells regularly. That's about 400 microliters of volume, and you're already well within the tolerance and the acceptance range. So, well, again, this is just a phenomena for beads where we need to do this aggregation and division procedure. Essentially, the tool counts the right amount of material there, and it's very powerful. It's a very powerful tool to truly understand how much material do I have in there. And this was validated, right? So like I mentioned in one of the first slides, is that you can validate these results by manually counting what's in there. In the case of beads, we did a 4-tool inter-instrument experimental approach to this, and very much the results lied within error, right? So the tool is counting properly, and several tools count properly. One of the greatest advantages of the Aura is that you can actually sample the same time with multiple tools. For those of you who need more than one Aura in the same system, so you can basically qualify multiple systems at the same time. So this process was actually quite easy to do. And now moving on to some of the newer capabilities that we like to showcase is to really understand. So sure, you may have some particles, but what are they really, right? And we added some amazing fluorescence capabilities to do protein ID detection on the tool. And fundamentally, what the vision for the Aura ultimately is that not only do you know at the single particle level, and get their size and their morphology, get the particle size distributions, get these high level insights, but ultimately use Aura to understand that for all of these constructs, are you looking at these particles? Are they your drug product or are they something else? And that completely changes the way from the earliest stages of how you can optimize your formulation and debug what might be going on. And for that case, we're going to, a small case study where we work with ethylene tetrafluoroethylene. As many of you here might be familiar with, NIST has this plastic mimic that looks just like a protein aggregate. It has the same morphology and very low refractive index contrast. And there was this great article that came out of the Japanese Pharmacopoeia that they qualified these samples with flow imagers. And a quick test for you guys is, can you tell me which, there's two of these 3 sets of images are proteins and one of them is not. And if you guess that these 2 were the proteins, you have a much better eyesight and intuition than I have. So they're basically morphologically indistinguishable. You can't just use the fact that some are lighter or brighter or their morphology to identify them, just because they're simply not enough. They're very powerful tools, but for full ID, they're essentially just a surrogate and a helper for characterizing these particles. And at the opposite end of the spectrum, you have these low throughput Raman detection systems that are used very much forensically. Once you spent years optimizing something, but it broke, and then you're like, I have to figure out what's going on. And that's an incredibly arduous, expensive, laborious, and highly expert type process. And unfortunately, it takes forever, and it's not really intuitive. So in this case, if you see a 1556 wave number, that means that you have an amide-2 bond there, and therefore it's an indicator that the material is probably proteinaceous. So this is painful. I built Raman systems before, and there's better ways to do this. And that's where we come in. So we added the capabilities of supporting fluorescence in our workflows and basically revamped the software so that it's more intuitive and flexible and supports these workflows in ways that we simply couldn't do before. And we're now transforming these grayscale images to color images so that your proteinaceous particles are in red, and particles that are protruding out of plane in the case of high semi, high side illumination, which, by the way, is one of the light sources that are required by USP788 microscopy. We have that built in our system just from the ground up. And we have a small case study where we want to distinguish, again, between proteinaceous and non-proteinaceous samples where morphological filters aren't enough. The workflow essentially just adds a fluorescent dye that is very specific to protein aggregates. It's called thioflavin T, and it's been validated for decades as the primary dye to characterize the binding of protein aggregates. Essentially the protein aggregates tend to form these microfibril structures due to misfolded beta sheets, and THT is very specific to those and highly water soluble. So we just add an addition stain, which is in step 4, which is steps 1, 2, and 3 are essentially just your standard BMI approach. Then we add a dye, and then we remeasure. This workflow is also highly flexible, and you can basically do them all just at once, just like you do in Horizon, by applying the dye right after you apply the sample. But happy to talk to you about that if that comes up. And essentially the system works. It responsible to controls. The IgG aggregates are highly fluorescing. The blue signals that you see there, they're not really fluorescence. They're actually the semi-channel being detected as ETFE protrudes out of plane. So essentially ETFE doesn't stain, right, but it protrudes out of plane. So it's very different to proteins, while proteins highly fluoresce under THT, but they don't really show any blue, so they do not protrude out of plane. So needless to say, these particles couldn't be any more different, and with a flow imager and with traditional morphological techniques, you really don't have the basis for distinguishing those two. One simple way of distinguishing them is look for particles whose fluorescence is significantly above the background, and we have great methodologies for separating the two. And in this case, we show a mix of proteinaceous sample mixed with ETFE, and this tool highly predicted, using this THT approach, that basically the ETFE particles did not fluoresce above the threshold. 99.8% of the IgG was properly characterized, and then for the mixed samples, it basically said that around 88% of those samples fluoresced. And while that result of the 88% may be confusing, we ran a controlled experiment so we know how much ETFE and IgG went into these given wells, because we conducted a controlled experiment using serial filtration, and it was basically dead on. So what our fluorescence-based detection approach predicted for the protein-on-protein content was exactly right by this counts-based experiment, in which we ran as a control. So the approach works. THC is very specific to proteins and not specific to the non-proteins. Going back into this, right, just put a picture back of these ETFE particles and the proteins, and they were very indistinguishable. If we plot fluorescence versus side illumination, you can see that these particle populations couldn't possibly be any more different, right? And those are the kinds of very important insights that you can get, and you can only get with the Aura. This approach responds very well to negative controls, from polysorbate particles to steel particles, etc., or to even like particles that have delaminated from Corning cryovials, which tends to happen frequently, as we see in some of our customer sites. There's more to it than just protein-on-protein detection. We have more channels available for different applications. We're currently building them up, and we'd like to get your feedback. Lots of interest on lipophilic particle detection, but it's pretty flexible, and we like to hear what you have in mind. Some of our AAV customers have some ideas on the kinds of things that they'd like to analyze as well, for example. So we love to hear your thoughts, but very customizable to your needs. So to wrap up, Aura is compatible with USP788 as of February of this year. It's high throughput, low volume analysis, so you can expedite your workflows to now last only a few hours as opposed to a few weeks. If you want identification, is actually a few hours, because the time is essentially the same to run a fluorescence versus a non-fluorescence approach, versus a few months if you're using a Raman system. low sample consumption, high fidelity, 100% sampling efficiency. All your particles are in focus. 21 CFR compatible, and it's very easy to use. So with that, happy to take your questions, and thank you so much again for attending.
Thank you very much, Bernardo. Great webinar. And just as a reminder to everybody, if you have a question that you'd like to submit, please submit it through the question chat window down below. And we'll go ahead and start. We got a number of questions that have come in. I will go ahead and read them off to Bernardo. And the first question is, have we compared the Aura to manual membrane microscopy?
Yes, we compared the Aura to manual membrane microscopy using bead standards. We compared it over 3 users and 4 different auras, and basically the prediction is that it's highly compatible with manual membrane microscopy. If anything, the results of the Aura were less user dependent, and the workflows were very fast. So for example, to count 150 particles in the membrane, as you would get with a 3,000 particles per mil solution, it took, and we're now looking at discrete particles, right? So it's as easy as experiment as you can possibly run. It took 5 to 10 minutes a well, and it's pretty exhausting, right? So we managed to do that with the Aura and show that the results not only correlate, but they're better and they're much faster.
Okay, great. Thanks, Bernardo. The next question, just to follow along the USP line of questioning, is what membrane pore size is acceptable for USP testing?
So the membrane pore sizes that are set forth in the chapter are basically just below 1 micrometer is what's recommended for USP testing. Our default membrane size is 400 nanometers, which is well within that range. And in the new chapter 1788, they make it perfectly clear that you can use more than 1 membrane type. So the reason why the original membranes were these color contrast membranes is because they relied on manual by eye type detection. But understanding that these new automated digital technologies such as this are now around, those requirements have also been relaxed, and our membranes definitely qualify.
Okay, great. Another question that came in is, do we detect silicon oil?
It's a good question, and the short answer is no. Silicon oil tends to flow through the membrane. It's been published by this paper that I referenced from Coriolis, as well as our own early insights. I think that the jury on silicon oil essentially is out in that it's not immunogenic. So while I can understand why some people want to characterize it, plenty of our users have also said that it's good that we're not measuring things that can confound the measurements, particularly in the small particle size ranges. So in short, we do not measure silicon oil. It goes right through, and we've actually received quite a good reception as a result of that, because they tend to confound measurements in the smaller bins.
Okay, great. Thank you very much. And another question is, do we have CFR Part 11 software?
Yes, we do.
Great, thank you very much. Another question is, do we know if there's a viscosity cutoff for the membrane filtration step?
I think we have it set at 50 CPs, but we've, from a more practical perspective, we have customers working with formulations with concentrations up to 200 mg per mil, highly viscous samples. And if you have any interest in that, we'd be more than happy to. We have a tech note that we put out maybe about a month ago or 2 months ago that talks about what are some of the suggested workflows for dealing with highly concentrated protein samples.
Great, and that's available for download on our website. Another question along the lines of filtration is, what is the vacuum pressure that we typically recommend?
I believe that the default vacuum pressure is 45 kilopascals, which is almost about a little bit under half an atmosphere. So you have the arrow pointing at around 85 degrees in your on the regulator, but 45 kilopascals.
Yep. Great, thank you very much. And there was a question about AAV. So I think to be a little bit more specific on this question is, can you talk a little bit about, does AAV particles, are they measured with the aura, and do they stain with thioflavin T?
Yes, so we can measure them with the aura. We can measure them for different serotypes with volumes as low as 10 microliters. We could probably even go lower. And yes, they do stain with thioflavin T, and we've even shown that we can distinguish those with non-proteins also present in those formulations.
Great. And we actually have an application note coming out on that topic very shortly, probably in the next couple of weeks. So check our website. That'll be available for download in not too long a time. Percent variability. Do we have an expected percent variability that we would expect from a typical sample?
It's a good question. It's highly sample dependent, but for example, when we come to bead standards, right, we measure them within the 10% threshold. I think that for thermally generated aggregates, typically the CVs are below 10%. That's sufficient particle population, say above 20,000 per mil. But again, it's a very important, more than instrument dependence, right? So like the inter-instrument variability is much, much lower than 3%. So most of the times, I think it's important to realize that the variability isn't necessarily on the instrument, it's actually on the samples, right? And as you can see, sometimes even when you run lots of volume and sample, that the CV essentially stays the same. So that's an inherent characteristic of these samples as they go through. So it just depends. The short answer is that it depends on the sample.
Okay, great. And one last question is dimers and trimers of proteins, and do we measure those? Are they large enough typically to be measured on the aura?
Good question. No, those are that's more on the SEC domain where you're looking at less than, like 10 to 20 nanometers. We're more on the 1 micrometer and above, focusing on the subvisible size range for this protein line, for this product line.
Okay, great. And then another question just came in. Is there a maximum particle count for the aura? I guess that goes to the linear range question. So what's the, the maximum particle counts, essentially what is the end of the linear range for the aura?
We've measured so concentrations up to 3 million particles per mil for very small particle sizes, and so about 3 million per mil for particle sizes on the order of about 2 microns. And again, this question is, and not to give a long-winded answer, but it's also sample dependent, right? If your sample has lots of large particulates, that changes this equation. But for model samples that are small, 3 million particles per mil.
Great, thank you very much. Okay, I think that's all the questions. And if there's any questions that I possibly missed, we can certainly answer those offline. Or if you have additional questions that you think of later, you can email them into Halo Labs or submit them through our website. Thank you very much, Bernardo. And that concludes our webinar for today.
Thanks, everybody.